So let's study sexual reproduction in flowering plants. Uh, you know the structure of flower already, and inside the flower the male and female gametophyte is going to develop. So let's study. Male part of the plant is this anther. This anther is bilobed. It has two lobes. You can see the two lobes. Bilobed anther is attached with a filament. These two lobes, actually they have four chambers in between. If you cut it like this, you will see the four thecas, or it is dithecus on one side. So the pollen grains are produced inside this pollen. There is sporogenous mass in the beginning, and when this is going to dehisce, means when the anther is going to break, sometimes when you touch the anther with your hands, the yellow mass comes on your hand. तो वो जो yellow mass है, that is pollens. So the pollens, they come through these line of dehiscence. They are germ pores, we'll see later. So the pollen is going to come out of the anther. So this is the line of dehiscence. So the let's study the detailed structure of this anther. The anther has, this is the anther. This anther has layers, some external layers. The outermost layer is called the epidermis. So you know the in plants outermost layer is epidermis. Below this there is a layer called endothecium. The endothelial cells, they are going to develop some kind of thickening material at maturity, and they help in dehiscence. जब dehiscence का time आता है, इसके endothelial thick, ये इसके अंदर कुछ ड्राइनेस सी हो जाती है, which helps in breaking of the anther. So then next you can see two three middle layers, and innermost layer is called the tapetum. This tapetum is the nutritive layer. This provides nutrition to the growing pollens. Pollens are going to develop from these mother cells, which are microspore mother cells. These microspore mother cells are diploid. They will undergo meiosis to produce, uh, later on they will undergo meiosis and produce the haploid spores. The haploid spores are formed in the form of tetrads. This is the mature anther. So this mature anther has pollen grains. These pollen grains are in the form of tetrads. Tetrads means four pollens are joined together. Three you can see here. The other one, the fourth one will be at the back. So these four tetrads in the form of these pollens will be released in the air. After this, they will detach. This one pollen grain will detach from here. So these pollen grains, now these pollen grains, these are the tetrads, and these pollen grains, the structure of the pollen grain we'll study in detail. Before that, you can see here there is germ pore. There is germ pore here. This germ pore is the place from where the pollen tube is going to come out. When the pollen is going to germinate, so the pollen tube will come out. So you can see furrows also. These are the furrows. These furrows are the unthickened areas in the thick exine. This is the exine of the pollen. Let's study the structure of pollen grain or the female gametophyte. Now this is the mature pollen at a two-celled stage. This is the two-celled stage. You can see there is a large cell. What are pollen grains? Pollen grains are male gametophytes, or the male gamete carrying. So they have developed from microspore mother cells. After meiosis, the tetrads were formed, and then they are formed. Now the nucleus divides into two. The two nuclei are produced. There is a cytoplasm around this nucleus, and this nucleus is also having a cytoplasm. First it is attached, but later on it is detached and it is suspended inside this cytoplasm. So this nucleus is surrounded by a little cytoplasm, you can see, and this cell is called the generative cell. This generative cell is going to produce male gametes, two male gametes. In case of angiosperms, two male gametes will be produced. We'll study later during fertilization what happens to those two male gametes. So this is the generative nucleus and cytoplasm together, it is the generative cell. And this larger one is the vegetative cell. It is the vegetative nucleus, and this whole cell is called the vegetative cell. This will help, this cell produces the pollen tube. When the pollen is going to germinate on the stigma, it will produce the pollen tube. This is the germ pore. This is the external wall of the pollen, which is very thick. It is called the exine. An inner layer which is made up of pectins and celluloses is called the intine. Exine is made up of a very resistant substance called sporopollenin. Sporopollenin, one mark question. Most of the examiner, they are very fond of asking this question. Sporopollenin is the resistant. It makes the pollen resistant to various temperatures, acids, and so many adverse conditions of the atmosphere. So this exine is the decorative wall of the pollen. We'll see in the next slide. It's so beautiful. It has some unthickened areas, but it is very thick protective layer made up of sporopollen. Now the uh, pollen grains, you can see they are so beautiful structures of the pollen. Exine is so beautiful, and here you can see the furrows, and these are the furrows. So these are the points from where the pollen tube or germination of the pollen is taking place. Now nowadays the pollen, since it is very nutritive, is being used as a food also. So this pollen is being eaten in some countries. Now we'll study the development of female gametophyte. So female gametophyte develops inside the pistil. This is the pistil. This portion is called the stigma, then style, and this is the ovary. Inside the ovary you will see the production of ovules. These ovules inside the ovary, this was the external structure. Inside there are ovules. So this is the ovule, structure of ovule, and inside the ovule is future seed. So inside the ovule, the female gametophyte is going to develop. The egg is going to develop. So let's study these two outer and inner integuments. Integument means uh, skin. These are the two outer layers of the ovule. They are called the skin of, they are the skin of the seed. You know, they will in future form the skin of the seed. The testa and tegmen of the seed, you must have seen in gram seed, it is brownish in color. So and the inner is transparent and whitish in color. In pea you must have seen sometimes it, you can see the outer layer coming out in your vegetable. So uh, in your soup, this area is called the nucellus. This is the tissue is called the nucellus tissue. Okay, this end of the ovule is called the chalazal end. This is the chalazal pole. So and this end is called the micropyle. Micropyle is the you can you must observe today only, go and see in your kitchen. This micropyle is the pore at the top of a gram seed. You can see small pore. This is the point from where the pollen tube is going to enter. The pollen tube or the male gamete carrying tube will enter through here to fertilize this egg. Now in the young anther, this nucellular tissue, this tissue this tissue has certain cells like this, and these cells, one of the cell will act as the mother cell. Okay, that mother cell is going to produce this embryo sac. These eight, you can see 8 nuclei and little at in the embryo sac. This whole is called the embryo sac. So these 8, how they are formed, let's study. Here you can see the single cell which is going to produce those 8 is called the megaspore mother cell. This megaspore mother cell is going to divide by meiosis. This is diploid. Okay, so this is going to divide and produce 4 megaspore tetrads like microspore tetrads. These are also megaspore tetrads. But here, out of these 4, 3 are going to degenerate. A typical plant study is being studied here. Variations are always there. Biology is a science of exceptions. Kahin na kahin kuch na kuch variations you will see, but this is a typical case. 3 will degenerate and 1 is functional. This functional megaspore is going to divide and redivide and to produce from one nucleus 8 nuclei are produced. Okay, 2 nuclei will come in the center. These are called polar nuclei, and later on they will join to form a central cell. Okay, this here towards the micropylal end, the central one is called the egg, and 2 surroundings are called the synergids. So egg and synergids together form the egg apparatus. Egg apparatus. So this this is egg apparatus. These 2 polar nuclei, later on they will form a single cell, and these 3 are antipodals towards the chelysal end. These are the antipodals. So this whole is the embryo sac. Here you will see the egg or the female gamete. So this is the development of female gametophyte. You must have understood completely. Now the mature ovule. This is the picture from your book, so must have seen. This is a mature embryo sac. This is the anatropous ovule, a mature anatropous ovule. Here you can see a diagram drawn by some artist. This is the chelysal end, and this is not nucleus, this is nucellus tissue. I have already told you, nucellus. Okay, these are antipodals, embryo sac, and secondary nucleus. Here you can see at maturity, those polar nuclei, they have fused together to produce a secondary nucleus. Can you tell me what will be the ploidy level? It will be diploid, because 2 haploid, they have nucleus have joined, so it has become diploid. We'll study later what happens to this secondary nucleus. Now here you can see the egg apparatus, the egg and the synergids. Okay, 2 integuments, and then this micropyle. The point where this funicle is the stalk with which the ovule is attached on the placentae of the ovary. This point is called the hilum, where funicle is attached with the ovule. And this is a ovule which is inverted, anatropous. Now I will show you another ovule which is not, which is upright, which is orthotropous ovule, to just to compare, because I have seen so many times the structure of ovule is asked in the exam, 3 marks question. Draw the structure of an anatropous ovule. So that is the L.S.OF an anatropy, longitudinal section of an anatropous ovule. This is orthotropous, just to compare. Always draw anatropous ovule. So this is all about the development of female and male video. So male and female gametophyte development you have studied. Please subscribe and share my video. Now we'll study this process with the help of some different So let's study the structure of anther, which is the male part of the flower. The anther has two lobes. These two lobes, bilobed anther it is. From outside they are seen as bi, but actually inside there are four lobes like this. Means the anther has four lobes like this. Okay, and in each lobe there are two lobes inside each lobe. So this kind of anther is called diathecous anther. They are joined together by a connective tissue. 2-3 layers layers which are covering layers of the anther. Let's draw the anther here. This portion, only this portion will be magnified here. The outermost layer is called the epidermis. Let's draw the epidermis. I draw myself so that you can see the diagram, but you have to draw in the exam. So for a drawing purpose you must learn the drawing part also. So that is why मैं बनाती हूँ खुद. तो ये this layer below the epidermis is endothelial layer. Endothelium is going to help during the dehiscence of the anther. जब anther की dehiscence होगी तब ये उसको काम आएगी. उसके बाद 2-3 layers हैं which are called middle layers. तो 2-3 layers are below this. So draw करके ये 2-3 layers middle layers. outermost layer भी लेबल कर देते हैं epidermis. Okay. Innermost is एक nutritive layer है which is called the tapetum. ये जो 2-3 middle layers के नीचे जो सबसे innermost layer है that is the nutritive layer. ये nutrition देने वाली है growing microspore mother cells. Now यहाँ पे you will see inside जो हमने अभी बनाया वहाँ पे that is the sporogenous mass in the young anther, which will have this sporogenous mass which will have a diploid nucleus. These are diploid nuclei. These diploid nuclei containing cells, they are called the microspore mother cells. These are diploid. These microspore mother cells, they will divide by meiosis. They will divide by meiosis since they are diploid to produce tetrads like this. Tetrad microspores, each of these is going to become a pollen grain. Okay, the tetrad में 3 you can see here at the front, 1 will be at the back. So let's study the structure of pollen grain in detail. The pollen grain has the outermost layer. So first I will draw the tetrad only. Well, let's see, ये ऐसे ही बना देते हैं पूरा tetrad. Okay, now this tetrad will detach to produce one pollen grain. Now this pollen grain, structure of pollen grain, it has very thick outer layer which is called the exine. Exine, like this, draw like this, but at some points I have not drawn this exine. These portions are called the germ pores. These are called the germ pores. From these points the pollen tube is going to come out when this pollen is going to germinate. Okay, so inside I'm drawing a section of the pollen grain from inside. This is called the intine. Before that, let's discuss about exine. Exine is a beautiful textured layer. It is made up of a substance called sporopollenin. Sporopollenin is the most resistant substance on this earth. जो pollen grains हैं वो दुनिया की सारी adverse conditions को face करते हैं, गर्मी, सर्दी, rainfall, acids. सो उनको प्रोटेक्ट करने के लिए ये स्पोरोपोलिन उनको प्रोटेक्ट करती है. इट्स अ वेरी रेजिस्टेंट काइंड ऑफ सब्सटेंस. सो एक नंबर का क्वेश्चन आता है स्पोरोपोलिन के ऊपर. देन देयर इज इंटाइन व्हिच इज मेड अप ऑफ सेलुलोज एंड पेक्टिन. ओके, so this is made up of cellulose and pectin. Inside, this nucleus is going to divide and produce a smaller and a larger cell. The larger cell is called the vegetative cell and the smaller cell is called the generative cell. Generative cell. This generative cell is here. You can see I have made it attached to it. These are the vacuoles. But later on, this generative cell will detach and it will float like this. So must have seen some diagram. If you have seen this kind of diagram, so this generative cell and generative nucleus. This is the generative nucleus. So exine and some vacuoles. This is the final structure of the pollen grain when it is released in there at a two-celled stage. Okay, here this pollen grain, this generative nucleus, and is going to divide and produce two male gametes. When this pollen is germinating on the stigma, so it will become three-celled when there is pollen grain germinates. Okay, not in the beginning. So this is all about the development of male gametophyte. So clear. Now we study the development of female gametophyte. Before we study the developmental stages, let's study the structure of an anatropous ovule. एग्जाम में आपको बहुत बार पूछा जाता है एनाट्रोपस ओव्यूल के बारे में. सो एनाट्रोपस ओव्यूल का डायग्राम तो एग्जाम में आता ही है, 3 मार्क्स का. आई हैव सीन सो मेनी टाइम्स. सो उसको ड्रॉ करना आना चाहिए तुम्हें. तो उसको ड्रॉ करो. इसमें there are two layers, the outer layer and then there is an inner layer. These are covering layers. इसकी 2 covering layers होती हैं और ये covering layers बाद में सीड की कोट्स बन जाती है. सीड कोट्स यू नो चने का दाना देखो. आज ही जाके किचन में आप चने का दाना देखना तो तुम देखोगे कि जो चने का दाना है वो उस ऊपर से तो ब्राउन है और अंदर की तरफ से वो थोड़ा वाइट कलर का है। सो जो वाइट कलर की है वो इनर इंटेगुमेंट है। इंटेगुमेंट का मतलब स्किन है। ओके, सो आउटर एंड इनर इंटेगुमेंट, दो लेयर्स होती है। ये सारा टिश्यू जो अंदर है इट इज कॉल्ड द न्यूसेलस। इस न्यूसेलस टिश्यू में एक आधा सेल जो है वो ही मेगास्पोर मदर सेल बनने वाला है, वो हम आगे पढ़ेंगे। पर मैं इस वक्त मैच्योर एनाट्रोपस ओव्यूल का डायग्राम बना रही हूँ। फाइनली क्या होने वाला वो ये, इस मैच्योर एनाट्रोपस ओव्यूल में आप लोगों को अगर ड्रॉ करना आता है तो आप लोगों ने ये पूरा बनाना है ऐसा। ये इस एम्ब्रियो सैक में, दिस इज द एम्ब्रियो सैक में सेंटर में देयर आर टू पोलर न्यूक्लियाई। Here towards a nucleus, these are called the antipodals. ये जो एंड है इधर, this end is called the chalazal end. ओके, और ये वाला एंड जो इस तरफ है दिस इज कॉल्ड दी माइक्रोपाइल। तुमने चने का दाना आज अगर ध्यान से देखोगे तो तुम्हें उसके ऊपर एक छेद नजर आएगा छोटा सा, दैट इज द माइक्रोपाइल। माइक्रोपाइल वो पॉइंट है जहां से पोलन ट्यूब इस ओव्यूल के अंदर एंटर करेगी। ओके, यहाँ से पोलन ट्यूब एंटर करेगी। ये सेंटर में इज एग, और इसके आसपास दो सेल्स जो दिख रहे हैं आपको, दे आर कॉल्ड दी सिनर्जिड्स। तो एग और सिनर्जिड्स दोनों मिला के बनाते हैं एग एपरेटस। तो एग एपरेटस ये है फीमेल गैमीट। ओके, ये जो सेंटर में दो पोलर न्यूक्लियस हैं, दो न्यूक्लियाई हैं, ये बाद में मिलके एक न्यूक्लियस बना लेंगे विच इज कॉल्ड दी सेकेंडरी न्यूक्लियस। वो हम बाद में पढ़ेंगे कि उसका क्या रोल है सेकेंडरी न्यूक्लियस। तो कई बुक्स में अगर एक ही दिखाया हो, यहाँ सेंटर में एक सेल दिखाया हो, तो दैट विल बी डिप्लॉयड विच इज ए सेकेंडरी न्यूक्लियस। तो अभी दो पोलर न्यूक्लियर दिखाए, दोनों हैप्लॉयड। सो और ये जो जिसके साथ ये फ्यूनिकुलस, वो डंडी है जिसके साथ ये अटैच्ड होता है। तो ये फ्यूनिकुलस इज द portion with which it is attached to the funiculus। ये और ये हाइफी जहां से ये इसके साथ हाइलम, सॉरी, ये फ्यूनिकुलस इज अटैच्ड टू द ओव्यूल, उस पोर्शन को कहते हैं हाइलम। So this is all about the structure of ovule. Now, development of female gametophyte. Ovule के अंदर one of the cell is going to act as megaspore mother cell। इसके आसपास वाले सेल जो न्यूसेलस हमने पढ़ा अभी, वो न्यूसेलस वाले सेल्स में से one of the cell is acting as a megaspore mother cell। Okay, this megaspore mother cell will divide and redivide to produce, it will divide by meiosis to produce 4 cells. Out of these 4, 3 will degenerate means खत्म हो जाएंगे, and 1 will be the functional. This is the functional megaspore mother cell. This functional megaspore mother cell nucleus will again divide to produce 8, 1, 2, 3, 4 and 5, 6, 7, 8 nuclei. So that will form the embryo sac. 3 will go towards one side, 3 will go towards the other side. They are called the antipodals, and 2 will be in the center. एक typical plant के बारे में बात कर रहे हैं. बायोलॉजी एक एक्सेप्शंस की साइंस है, तो कुछ ना कुछ अलग भी हो सकता है किसी में. तो एग और सिनर्जिड्स और ये जो है एंटीपोडल्स. तो ये है इसकी फीमेल गैमेटोफाइट की डेवलपमेंट. यानी दिस फीमेल गैमीट एग will be fertilized by the male gamete produced in the pollen. तो ये पोलर न्यूक्लियाई है. तो दिस इज द फीमेल gametophyte development.